Okay, class, we're ready to talk about something of major importance in cell activity, known as protein synthesis. This will be divided into 2 or possibly 3 videos. This is so important. All right, let's start. DNA is the master blueprint for protein synthesis and RNA synthesis. Okay, remember that the only thing from a previous video that DNA does directly is to code for the primary structure of a polypeptide or protein. Let me say it again. The only thing DNA does directly is to code for the primary structure of a polypeptide or a protein. On DNA, we have genes. A gene codes for one polypeptide. It used to be said a gene coded for a protein, but some proteins are in the quaternary structure, which means the protein has more than one polypeptide, like hemoglobin. So now it's one gene, one polypeptides. Within a gene, you have triplets. Triplets of nitrogenous bases, or should I say nucleotides, that are the code for this. So we'll be looking at that. Okay. Each triplet on the DNA codes for one amino acid. Each triplet codes for one amino acid. Remember, we had 20 naturally occurring amino acids. Okay, let's go further. We divided our cells in our body into somatic cells. We said we had around 210 different cell types. About 100 trillion cells, but about 210 different cell types. And we had divided the cells into 2 types, somatic cells and gametes. Somatic cells were all the cells of the body except the sperm and egg. The somatic cells had 23 pairs of genetic material, 46 pieces. The gametes only had half of that, because when sperm met egg, fertilization would occur to restore the 46. Okay? We said that a genome is a complete set of genes in any organism. A cow has a genome, a mosquito has a genome, but our human genome has 23 pairs of genetic material. 22 of these are called autosomes. The 23rd pair are the sex chromosomes. The 23rd pair are the sex chromosomes. Okay? So when we look at that, if you recall, we'll go a little further. We look at that, we had this thing we call a karyotype I mentioned. And I said that's how we would lay out the genetic material. Let's say if you had an amniocentesis, they would extract the DNA and they could lay it out in this pattern. This is chromosome pair 1. The chromosomes are determined, each chromosome is determined by its length, its centromere position, we'll talk about, and how it stains. In the official layout of your human genome, the first 22 pairs officially laid out are the autosomes, and the 23rd pair they've relegated in the official pattern to be the sex chromosomes. This is called a karyotype. Typing the caryon again was the nucleus. Okay? In protein synthesis, the major players, not the only players, are going to be DNA, deoxyribonucleic acid, RNA, ribonucleic acid, and the 3 main types, not the only, you hear me? Not the only, but the 3 main types are mRNA, R, and transfer. They stand for messenger, ribosomal, and transfer, that we'll see. Okay? Reviewing. The ribosome, which we talked about in a previous video, has 2 subunits, a small and a large. These stay separate. They float in the cytoplasm separate until protein synthesis is to occur. When protein synthesis occurs, then they hook together. Now, when they hook together, if a protein is to be used inside the cell, is made on free ribosomes. If a protein is to be used in the cell membrane or exported out of the cell for exocytosis, it's made on a fixed ribosome, and the fixed ribosome is attached to the endoplasmic reticulum. This is material we'd already discussed in a previous video. Okay. We have a polypeptide coding gene. Remember, one gene, one polypeptide. That's a region of DNA that codes for one polypeptide. A DNA reading frame. This is kind of like, when you look at a book and you're reading, your eyes get a few words and then they move over to another few words. So think of that as a reading frame. A reading frame on DNA are regions within the gene that code for one amino acid, which would be a triplet code. Regions within a gene, that's what it concentrates at one time. Because remember, DNA codes for the primary structure. That's all it does directly. And the primary structure is the linear sequence of amino acids. So you're reading and putting an amino acid. It's reading and putting another one. Reading and putting another. A DNA reading frame contains 3 nucleotides. Remember, a nucleotide is the monomeric unit of DNA and RNA. It consisted of a nitrogenous base, a sugar, and a phosphate group. When it reads, there's an enzyme that's going to read it, and it's going to read it in the 3 to 5 direction, the 3 to 5. Now think about this. You're reading this one I'm showing you in a certain direction. We don't read backwards, we read forward. So the enzyme does the same thing. Thus, if a polypeptide had 100 amino acids, the DNA would need minimally, hear my word minimally, 100 reading frames, because 3 would be for each amino acid. So you'd need 300. Okay? So kind of study that. Okay? Now, the DNA hands its signal to RNA. The DNA hands its signal to RNA. All right, here's my analogy of that. You have a cell and you have the blueprint inside the nucleus. All right. You make the protein in the cytoplasm, not in the nucleus. So you need to export the message to the cytoplasm to make the protein. Now, think about this. Let's say General Motors. Let's say General Motors in Detroit has one blueprint for a certain car, but it's going to make it in Texas, at a plant in Texas. If you took the original blueprint and sent it to Texas, they may never see it again. It may get stolen, lost. So what they would do is make a copy and send it to Texas. Make a copy and send it to Texas. So messenger RNA, that's where is carrying the message of DNA. What will happen is, as we'll see, is that messenger RNA will be formed as a reverse replicate of the DNA, so you can take the messenger RNA out of the nucleus and take it into the cytoplasm where the protein or polypeptide is going to be made. Now, when you take the message out there and you're constructing the polypeptide or protein, what's it constructed of? Amino acids. Amino acids are sitting or floating free in your cytoplasm. You keep an abundance of them in your cytoplasm, hopefully, in a good cell. And you have this construction job going on. So you need, for the message, to send a signal out to pick up the amino acids in the cytoplasm and bring them to the construction site. That is the job of transfer RNA. Transfer RNA's job is to bring the amino acids to the construction site. Now, how does it know which ones to do? Because the transfer RNA, we'll see, is reading from the messenger RNA. So let's think about this. You got the DNA, which reads into the messenger RNA. The messenger RNA then reads into the transfer RNA. So it gets confusing. So they had to come up with terms. So you have a triplet code on a triplet code reading frame on the DNA. The triplet code on the DNA then is formed into like a Xerox copy, but an inverse copy. You'll see what I'm talking about, an inverse copy into the messenger RNA. So now the messenger RNA is called a codon. So let's go back. The DNA is a reading frame triplet code. That's its code for one amino acid. Reading frame triplet code. On the RNA, the code for one amino acid, which it read from the DNA, is going to be called a codon. So a codon is on the messenger RNA. That's the term for that triplet code on the messenger RNA. And then transfer RNA is going to read from the messenger RNA. So it's going to read an inverse to that, and that would be the anticodon. So DNA, reading frame triplet code, RNA, codons, transfer RNA, anticodons, but all reading the same thing, but in the inverse, as you'll see. All right, so we go further. Here's so we got a genetic code on the DNA, and it's represented by a triplet code. The codon, though, is on messenger RNA. So what you're seeing here is the codon on messenger RNA, which was read from the DNA. Now remember, RNA does not have thymine, it has uracil. Now, let's say this. You have 20 naturally occurring amino acids. You have 20 naturally occurring amino acids. The RNA code, which ultimately came from DNA, has specificity but not redundancy. All right, now let me tell you what I'm saying. This is an amino acid, phenylalanine. There's another one, leucine. A codon has a triplet code too. So uracil, 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 or uracil, uracil, cytosine codes for phenylalanine. Now, try to think of this in a mathematical way. You have 3 possible spots here. Okay, in RNA, you have 4 possible nitrogenous bases. 4 possible. Adenine, cytosine, guanine, and uracil. Adenine, guanine, cytosine, and uracil. In all of these 3 spots, you could have any one of those 4 in that first spot. Any one of those 4 in the second spot. Because you could, just like here, just because you had this one in the first doesn't mean you can't have it in the next. All right. So if we go to math statistics, you could have 4 in the first spot, 4 in the second spot possible, 4 in the third. So then how many possible combinations could you have? 4 times 4 times 4. 4 times 4 times 4. Because you could have 1 of 4 here, 1 of 4 here, 1 of 4 here. I want you to think on that. That means you have 64 possible combinations that you could have. 64 possible codon combinations. But you only have 20 amino acids. You only have 20 amino acids. So the statement is you have redundancy but no ambiguity. Redundancy but no ambiguity, which it means more than one codon can code for the same amino acids, but none of these 2 can code for another one. So you can have redundancy, because you can have redundancy, but it's not ambiguous. UUU doesn't code for leucine today and phenylalanine tomorrow. Okay, so we're going to pick this up on the next one again in a moment.